Dr. Croce and Vinay and team. It was really, you had to twist our arm to get us to Whistler here. So thank you for all your efforts. And also all of you who thought this morning about putting an extra snooze and are here. Thank you. That's always first session genetics. So my talk, I was told to talk about how do we bring this into the clinic? How do we bring genome sequencing to patients? And for me, that means how is this useful for patients? And what have we learned over the past several years? And this is my usual disclosures. You can read them if you want. I'm going to say that I think it's proven to be a very challenging time for thinking about how do we bring genomics into the clinic. There's no doubt some examples in oncology where genome sequencing has dramatically transformed what we think about disease. And I'm going to use a very local example. I'm just talking to Dr. Trion outside, and you'll hear more from him about his discoveries in Waldenstrom's, where truly genome sequencing has the power to alter the natural history of the disease, where the therapeutics are going to make a difference. The unfortunate reality is that for most other tumors, I think we have an uphill battle. As you know, this is a classic slide from LEAF where we talk about the ways that we catalog tumors, the very elegant TC classification where we talk about 7, 8, and this is not unique to myeloma. As you know, this occurs in all cancers. But the reality is that there's many, many more. And in fact, recent work in breast cancer shows us that every single breast cancer is different. And we know that for myeloma, we know that for CLL, we know that for every single heme malignancy. And the reality is, how do we bring that complexity, how do we bring that heterogeneity into something that allows us to develop therapeutic strategies for? Now, I'll close by saying I think it's only in those diseases where you have a certain level of stability and more homogeneity that silver bullet-like strategies will actually pan out, as they have done so with imatinib in the case of chronic myelogenous leukemia. So I know some of you in the myeloma field have seen the slides before, but given the broader aspect of our audience, I'd like to just share with you work that our team at large, led by Dr. Stuart Bergsagle and with work from my lab as well too, have done, trying to understand this clonal heterogeneity within myeloma. We've had an opportunity to collect serial samples on a patient of ours who was treated with a number of regimens. As you can see, the patient is first treated with lenalidomide low-dose dexamethasone, experiences a relapse, goes on to get carfilzomib, and then a number of other series of treatments, each one of them with shorter duration of benefit. And what you see plotted there, of course, is the IgA levels in red, and then ultimately the free light chain, which escapes the last form of treatment and the patient succumbs to the disease. And then we had the ability of collect samples at multiple time points. And if you look at the genome-wide view on this, of course, you know, this is the horizontal depiction of array CGH. What you see is that there's chromosome 1, 2, 3, and 4, all the way to X and Y. And the genomes actually, at the broad level, look quite similar, with some differences, but for the most part, they look quite similar. But as has been said before, the devil's in the details. You start going to specific regions. Now it's presented vertically. You see that going to the left here, it represents there's a deletion, and in this particular case, it's a deletion that is maintained from the time of diagnosis until the time of relapse. Whereas on the right, you see evidence by CGH of deletion that was present there in the tip of chromosome 8, and then disappears at the time of relapse, and yet we see the appearance of a new deletion. Now, of course, chromosomes don't synthesize themselves. Again, you cannot repair the full length of an arm's chromosome. So the only possible explanation by just looking at this, which is proven subsequently, of course, is that this is a heterogeneous mixture of cells. Cells that don't have this deletion exist from the get-go. And in fact, those are the cells that predominate over time. And those same cells also have the deletion that's present underneath. So this actually provided us with the opportunity to go on and do single-cell analysis with FISH probes, just so that we could verify what was a component and how did those abnormalities relate to each other in the individual cells. Here's an example, of course, of a deletion of this gene BERK23 that becomes clearly much more apparent at the time of relapse with the presence of a biallelic deletion, and is depicted as well too in the right. So with this information, then we embarked on the single-cell analysis at the multiple time points. And this is our summary slide for those results. So as you can see, at the time of diagnosis, we identified 4 predominant subclones that are called 1.1, 1.2 in orange, 2.1 in green, and 2.2. Now, the little gray area that you see there is miscellaneous, represents the remaining subclones, and the number of them is vastly larger than those 4. In fact, we think it's probably thousands, if not hundreds of thousands of different subclones, each one with some slight variance, and each one of them competing for dominance for the tumor population. So as a patient goes on treatment, there's a remission. As you can see, the composition of the tumor remains quite the same, with seal clone 1.1 being predominant. But obviously, some of those clones adapt, they become resistant to lenalidomide, and now we have the emergence of clone 2.1, which is shown in green. The patient receives carfilzomib, the green is controlled, but we have reemergence of 1.2, and so on. And as you keep going, you see progressively growing for clone 2.2, which ultimately is the one that predominates over time, produces the free light chain, does not produce an IgA, and makes the patient succumb to the disease. And this, again, this is not unique to myeloma. I think this applies to every single human malignancy. It's just that we had the opportunity, because of the nature of the disease we deal with, to probe the patient at this multiple times. So this really poses a challenge, and I think the ability of tumors to have heterogeneity and to have subclones probably becomes the greatest challenge as we think about targeted therapeutics. And if we are to be successful, and this has been described in multiple ways, if we are to be successful, we really have to be able to control the majority of the subclones from the beginning, because there's evidence that human cancer can be cured, and this can happen again in somewhat stable situations, hairy cell leukemia, some of the childhood leukemias, but even sometimes solid tumors can be cured in the metastatic stage. Of course, the best example of this is adjuvant therapy for breast cancer. We must be able to control every single one of those subclones. So this is obviously just conceptual and hypothetical, but in my mind, that means that we have to go with combinatorial, aggressive upfront therapy for the disease, and I'm obviously open to the debate about that. Now I'm going to show you data from some of the very deep sequencing we've done, looking at subclone analysis. I'm not going to show you the data from the 2 papers that have been published looking at the whole genome in myeloma, seminal papers, which showed us, if anything, that heterogeneity continues to build and is greater than we have ever expected. But we wanted to ask a question, for instance, if we go with very deep sequencing, and this is work that one of our junior faculty did, Esteban Braggio, looking at the depth of P53. He has examples, for instance, where we can find this in a very tiny fraction of patients, where with subsequent follow-up of the patient, the mutant clones become the predominant clone in the patient. And again, this is just an example of a simple common gene that is associated with human malignancies, but would apply to any other gene that would dictate drug resistance or proliferation advantage. And in fact, once you apply this technique, you can see that it's not infrequent that you see very small subclones of the disease that will have this abnormality. This is very interesting because we haven't been able to formally prove yet the relationship of the mutation associated with the progression, because as you recall in myeloma, most of the cases that have the 17p13 deletion are associated with the non-mutated other allele, yet they have a very significant impact on prognosis. Perhaps this is the story, but I'll tell you, the first attempts we've had at this have not been successful in confirming that particular hypothesis. Now, my colleague, Dr. Stewart, who's sitting here in the back of the room, has taken the approach of saying, well, it's interesting to do the whole genome on the exome, but couldn't we get smarter and start developing panels for genes? So he's using, again, the ion torrent technique and has launched a research initiative where we're going to go do the targeted sequence of genes. And this will be obviously work in progress as that list will evolve as more and more genes are discovered or are implicated in the pathogenesis of the disease. And this is sort of a quicker turnaround time, which represents the same technology I just showed you for very, very deep sequencing. I think that the ability to solve this problem will not be in identifying the genes, but rather the ability to create the relationships and the linkage between those mutations and how that tells a story is really going to be how we get smart about approaching sequencing in the clinic. He's done validation for this, for instance, using tumors where there's a known sequencing through an exome, correlate this with the ion torrent, which you see on the left, so it really perfectly correlates with what we knew about those mutations. So I think the technology will be ready to test, again, known mutations in cases with multiple myeloma. It has a dilutional experiment there on the right, just again confirming the validity of this. Can do 244x. Of course, you can go higher depending on how much coverage you want to have. And of course, as we start running some of the samples, he's found that you find the predicted mutations. But again, the challenge will be how do each one of those mutations relate to each other to actually build the story of the pathogenesis of that very specific patient. Now at our center, we've also done the whole genome. This is part of an approach of trying to look for mutations in patients for whom there are no additional therapeutic options. This has been done in a number of tumors in conjunction with the Center for Individualized Medicine, and has clearly been a learning process. So this is one such example for patients with extramedullary myeloma, where the whole genome sequencing was done in the hopes, again, of finding actionable mutations. And it's probably not worth going into the detail, but I'll tell you some of the results are consistent with what you would see in the clinic. So the patient has cereblon mutation, cereblon being a key and essential, I would say, player in the whole way of response to IMiDs for myeloma patients. Mutations like NR3C1 potentially associated with resistance to corticosteroids, and also mutation in the proteasome subunits. Now, of course, this is a beautiful illustrative example, but the reality is when you do this, you don't really find cereblon mutation in the majority of patients who have IMiD resistance. There's many ways, and certainly more than one way, in which these mutations actually confer therapeutic resistance. And this is an example, again, just of the lanes with the mutation for cereblon. So, well, I'm basically personally an optimist. I'm somewhat just in shock of how are we going to bring the genomic sequencing into the clinic. And I want to believe we can do that, but I don't see a clear pathway, at least not right now, for multiple myeloma. Now, I love this paper, which was published from the Beijing Institute of Hematology, the Peking Institute of Hematology, where they actually looked at imatinib in CML, but now they didn't look at the chronic phase. They looked at the accelerated phase. And of course, those of us in myeloma don't read these papers, but when I saw this, I said, this is very, very illustrative. In the accelerated phase, imatinib is much worse than allotransplant, according to this particular study. So the question is, myeloma more like accelerated phase or like chronic phase CML? And I suspect it's probably more like accelerated phase. Now, obviously, we could find more targeted therapeutics, perhaps build on what we know from standard therapeutic strategies. You're going to hear more about that this afternoon. But we really have the question of how does that relate to actual myeloma. Now, I'll close just by showing you an example of further evidence of genomic instability, which leads to karyotypic complexity. This is a classic slide for breast cancer, where you see the lane in the center is a triple negative breast cancer, where you have what they call the stormy pattern, great number of deletions and mutations. So we took a similar approach in myeloma, using the same strategy, just using CGH, and published this paper last year in PLOS, where we actually show that if you just take measurements of genomic complexity as a surrogate of ongoing genomic instability, you can readily identify the most aggressive forms of myeloma, which leads me to believe now that perhaps as we move forward and as we continue to think about prognostic markers and even predictive markers, perhaps surrogate indicators of genomic instability or direct testing of genomic instability may represent the most important prognostic factor for the disease for multiple myeloma. And again, this is something we'll continue to work at. And I'm just going to propose that one potential way where we could get smart about this is actually to try to anticipate what comes next. And I've talked to a couple of people who have expertise in evolution and this nascent field of evolutionary medicine, saying that when you look at nature, you could almost anticipate there's a few predictable pathways where species are gonna vary to. If they're gonna survive, they're gonna be better off with evolution, and perhaps the tumors are the same way. And telling that story about the interaction between the different mutations may be the most important thing we can do. So if you have a patient with a RAS mutation, you probably can anticipate that the next 5 most common mutations might be this, and you try to anticipate that as we embark on smart therapeutic strategies. So with this, I'm going to close, and then thank you all for your attention again and your presence this morning.